Alright, let's get started. Today's study tip says flashcards should have a maximum of 15 words to be flashed. Study them back to front and front to back. So one of the things about a flashcard is that most students make them too long and you really can't flash them, right? So words and definitions are one of the best things to make with a flashcard. Word on the front, definition on the back. Now remember, study them from back to front because that's how I make the questions on Scantrons, right? On multiple choice. I usually put the definition, like, what is the bright yellow thing in the sky? And then I put, sun, moon, cloud, bird. I don't write what is the sun? A bright yellow thing in the sky, a fluffy white thing in the sky, an animal flying through the sky with wings. So think about how the test questions are worded. It's kind of like Jeopardy, I guess, right? You get the definition and then you have to find the word. Okay, so be careful on your flashcards. I like this little guy studying his kanji, having those rings but around the edges, to help you carry them. You don't lose them. Some of you don't like flashcards, and that's fine. If you haven't tried them, though, give them a try. See if they work for you. There's apps now on your phones that you can get to do flashcards. A lot of people like those, too. Go for it. Chapter 5, Cell Respiration and Metabolism. So we're going to go through metabolism of 3 nutrients. First one is carbohydrates. We're going to spend the most time today reviewing this. Then we'll talk about lipids, and then we'll talk about proteins. I expect you to already know all this stuff. So I think you have learned it either in anatomy or general bio. Several of you have taken microbiology. If you've never seen this before, then you're going to probably have to watch this lecture a couple times. Okay, so the 2 terms I need you to know. This is the word catabolism. Say catabolism. Okay, it's not, you don't accent the first A like catabolism, like cannibalism. Okay, it's catabolism, so you accent the second A, and it is the breakdown of nutrients for energy. We would say catabolic reaction, though. So you would say catabolic reaction, but you would say this word as catabolism. Okay, and this word you also accent the second A, so it's anabolism. It's the use of nutrients to build new products. You may have heard the word anabolic and like anabolic steroids, which are like testosterone, but this word is anabolism. Okay, say anabolism. All right, good. So carbohydrate catabolism takes up most of our time today, which, by the way, is a sign to you that it's a possible short answer question for the exam, right? Because it's worth a lot of my time, so therefore it's worth a lot of the test time, and a big thing that you need to know. So you already know glucose is a major energy source for the body. There are 3 steps in the carbohydrate catabolism. Say catabolism. First, you're going to go through glycolysis, and the goal of all these things is to make ATP. The citric acid cycle, some people call it the TCA or tricarboxylic acid cycle, and some people call it the Krebs cycle. I use the word citric acid cycle. Electron transport system, or the electron transport chain maybe it's called. Put together, you get glucose plus 6 oxygens will become 6 waters, 6 CO2s, 36 ATP, and heat. Now your book spends a good page talking about how you get the number 36 or 38, or I don't really care. Okay, so let that page in the book go. Just know that we're trying to make ATP. It's around 30 something. Okay, so people debate over it. I don't know. I got other things to do. Okay, heat is important to be made here. This is the same heat that keeps your body warm, keeps your body heat at a normal temperature. Okay, this is where the heat comes from. Of course, you get heat from moving muscles, right? Here's the first pathway, glycolysis. This is biochemistry. And when I was a biology student, this is something I had to memorize along with all of the structures. So here it just has the name. Like we start at glucose, the top, and we put in a little ATP. So we actually use ATP here to get glucose 6-phosphate. So I had to memorize the structure of what all these look like, the C's, the H's, and all that stuff. You don't have to do that. I do want you to look at this pathway though and have some take-home points, which will be on the next slide. Okay, so I want you to know an ATP goes in, another ATP goes in. Eventually, you're going to make two ATPs on the left, two ATPs on the right, so you make 4, but you're going to use 2. And you're going to take the glucose, break it down into 2 pyruvic acids, each with half the number of carbons, right? So we had carbon 6 in glucose, now we have 3 in each pyruvic acid. I want you to know that you do make some reduced coenzymes, so NAD becomes NADH on both sides. And I want you to know that this happens in the cytosol. Okay, it's not needing oxygen, so what's the word for not requiring oxygen? It's called anaerobic, right? So this is an anaerobic process. Okay, so glycolysis in the citric acid cycle takes glucose with 6 carbons, divides it into 2 pyruvic acids with 3 carbons. The coenzyme, becomes reduced. NAD picks up some hydrogens to become NADH and hydrogen. Pyruvic acid is made and it goes to the citric acid cycle, so we'll follow that next. The NADH and the hydrogen that goes with it goes on to the electron transport system. Okay, so on the test you're not going to write ETS for me. You're actually going to write the full term, electron transport system. I have it abbreviated here only because, like your flashcards, my slides are not supposed to have too many words. Otherwise they're too full and too small. So I abbreviate sometimes. 4 ATPs are made, 2 are used, and the final product is 2 pyruvic acid, 2 NADH, and a few ATP. Got it? Oh yeah, last thing. No oxygen is required, so this is an anaerobic process. This is a little reminder of how we're going to do this redox reaction, right? This reaction, I should have a little double arrow on the pink one here. This reaction is, if there is no oxygen, say in muscles or tissue, then the pyruvic acid, which normally goes to the electron transport system, has to be converted into lactic acid. So you know this, right? When you go jogging and you're not in good shape, you know the lack of oxygen means the pyruvic acid is converted to lactic acid and you get that burn, right? That's the burn in your legs and you're breathing. So what you have to do, of course, is stop and you rapidly breathe to increase the oxygen that comes into the muscles. That lactic acid then is converted back to pyruvic acid. So we learned about this in lab. Do you remember what LDH stands for? This is called lactate dehydrogenase. So this says if no oxygen, then the pyruvic acid is made into lactic acid and you only get 2 versus 30 or 30-something ATPs made this way. Okay, so not a very good deal because you only go through glycolysis, right? You never make it to the citric acid cycle and electron transport system. There's the LDH, lactic acid dehydrogenase or lactate dehydrogenase. Either word's fine. So let me ask you, going from left to right, here we go. Let's try it. What is reduced and what is oxidized? When you go from left to right, what is reduced? So remember, you get to choose from the reactants. If I ask you what is reduced as you go left to right, you get to choose either NADH or pyruvic acid. So we'll say what is reduced, because remember the oil rig thing? Reduced is to gain. So who's gaining when you go from left to right? Pyruvic acid is gaining. It's gaining hydrogens to become lactic acid. And NADH is losing, so NADH is being oxidized to become NAD. From right to left, you want to do it that way? Try it. What is being oxidized and reduced when you go from right to left? So reduced is gaining, so the NAD is being reduced to NADH, and lactic acid is being oxidized to pyruvic acid. Alright, give it a try. So the reaction is reversible, and we were reviewing this in lab, which organs take it which way. And you already know, muscle makes that lactic acid. Okay, citric acid cycle. Now remember the pyruvic acid had 3 carbons. Well, we're going to chop off one of the carbons, and add a piece to it called coenzyme A. And we call this acetyl coenzyme A. It says C2. So this is the pyruvic acid became acetyl coenzyme A. And then we're going to send it through these 8 steps of the citric acid cycle. Some of the key things that I want you to know. You don't have to memorize malic acid, fumaric, but remember last time we did succinic acid and fumaric acid? Let's see, I need you to know, what do I want? Some water being made, some water coming in. Here we go. An NAD becomes NADH. Is that reduced or oxidized? NAD is being reduced. CO2 comes out. I want you to know this. This is the carbon dioxide that you exhale every day. Okay, so I think the general public knows that we exhale carbon dioxide, right? But nobody knows where it originates from. And it comes from the citric acid cycle of every single one of your cells. And I think that's completely amazing. That all these little cells making CO2 is the same thing that you're exhaling. That's where it came from. It came from this cycle right here. Here's another one. Another reduced coenzyme. Some water coming in. Oh, there you go. 1 ATP. ADP to ATP. And another coenzyme called FAD, very similar to NAD, picks up some hydrogen. So this is reduced. And then let's see, going around. Let's see what I have in my notes. Oh, first of all, the location of where this happens. Just a reminder, the mitochondria has an outer membrane, inner membrane. The inside is called the lumen or the matrix. That's where this is happening. Okay, so it's in the lumen or the matrix of the mitochondria. The pyruvic acid becomes acetyl coenzyme A, which then goes into the citric acid cycle. 8 steps. You don't have to memorize the steps, just know there are 8. Also, no oxygen, so we say anaerobic. And you make 2 CO2s, 4 reduced coenzymes, and 1 ATP. So not really all about the ATP here. It's more about getting to the electron transport system, the 4 reduced coenzymes. This little guy in the corner, by the way, is not a peanut. He's supposed to represent what? It's a mitochondrion. Yeah, with his big biceps and his cool glasses. I like that guy. Now the next part is the electron transport system. It happens in the little finger-like projections that come through, called the crista. Okay, so over here is where we're going to have the next step. So, do you remember this study before? Have you done this? The outer membrane, the inner membrane, the space in between is called the intermembrane space. So what happens is all the reduced coenzymes at the bottom here, we have NADH. Do you see it? NADH is going to come up to this protein in the inner membrane. There's a second protein, a third protein, a fourth protein, a fifth protein. There's 5 of them. 3 of them are going to take the hydrogens from NAD or FADH2, either one, and pump the hydrogens into the inner membrane space. When the hydrogen comes off, the electron comes off at the same time. Okay, so that's the reason why the hydrogens go on and off, is because of electrons. The E negative, right, attracts the hydrogen, H plus. The hydrogens, if we follow the hydrogens, we see that they're pumped in through the second pump and the third pump. The membranes on either side are impermeable to hydrogens. What's impermeable mean? Impermeable means not permeable, okay, right? Not. So hydrogens cannot get through. So the concentration inside the intermembrane space starts to build up. So we get a high concentration of here, and they want to escape, okay, but they can't, except there's one exit, and that's this purple protein on the right side called ATP synthase. It's an enzyme, right? It's a protein that acts as an enzyme. You can tell by the name, ATP synthase, that it's going to do what? It's going to synthesize or make ATP, right? So as the hydrogens escape through here, yay, they get out. There is energy come from their flow that puts together ADP and phosphate to make ATP. Lots of ATP made here, okay? The electrons, on the other hand, they get moved from one protein. That's what this shape here is, proteins in the membrane, in the inner membrane. They get passed from one to the second, to the third, to the fourth, to the fifth one. At the fifth one, oxygen is put with hydrogen, and those electrons are used to make water. Okay, so the electron is passed, and then it's used to make water as a waste product. But this is a lot of water every day that's made, and your body actually, just reuses it. Okay, so we don't exhale the water like the CO2. All right, let me see what else I want you to know here. I think that's it. Let's go through the notes and see. So this is called the electron transport system. It's in the cristae of the mitochondria. Oh yeah, in the picture here, remember the third pump at the end, oxygen and hydrogens are put together. So since oxygen is involved in these reactions, this process is called aerobic. If you follow the hydrogens, you go to the pump, drop off the hydrogens from the reduced coenzyme, put them into the inner membrane space. That membrane, though, is impermeable to hydrogens. However, they can passively diffuse through the ATP synthase. And through the ATP synthase, as they flow, ADP and phosphate get together to make many ATPs. Following the electrons passed from one protein to the next one, and at the end it helps to put half of an oxygen with, well, we don't, not really half. It helps put O2 and H, but we have to balance the equation, right? So we write half O2 plus 2H plus to make water. Okay? Questions? Let me know. Oops, and we see it again. Here's your group question, or you're not in a group, unfortunately. You can discuss this, how about online with our class. These are meant to be hard, remember? And in the lecture on campus, I do have students work in a group, so I call these group questions. It says, some drugs inhibit the process of glucose metabolism. The result is a decrease in the production of ATP. Try to describe in cellular detail how the following poisons inhibit ATP formation in the mitochondria. First, cyanide. Have you heard of that? So this is stuff that would come with, a skull and crossbones on it. It inhibits the third pump, cytochrome oxidase. Second, rotenone. This is an insecticide to kill insects that they spray on plants. This is one of the reasons why it's good for you to eat organic. Okay, organic produce is not sprayed with this stuff. Rotenone blocks the NADH dehydrogenase. And third, there's a drug called FCCP. The real name is carbonyl, what a blah blah. Okay, and this drug makes holes in the mitochondrial membrane. It was given as a weight loss medication in the 70s. And people who were overweight took it. It put holes in the membranes of the mitochondria, and they lost weight, and then they died. Okay, so think about how that might help them lose weight, but actually causes a decrease in production of ATP. So all three of these, pause the show, go back to the picture, or I move forward to the picture. I think I put in a second one here. And think about how these things work to do that. Okay? Alright, so the first one, what's it say again? Cyanide inhibits the third pump, cytochrome oxidase. So if you inhibit the third pump, or any of the pumps, right? If you inhibit the pump, you don't get any hydrogens across, right? And if you inhibit this whole thing, it's kind of like a traffic jam, right? There's an accident. So will the electrons be passed? No, they won't. You can't pass an electron because it's all jammed up. You can't move a hydrogen. And if you can't pass an electron and move a hydrogen, you probably can't move the hydrogen and electrons here in the second pump, and you can't do it in the first pump. So you get no hydrogen concentration building up. You get no water, which means you don't get to do any of this. So the whole thing stops. Cyanide is an excellent poison. How about that? And it works by stopping production of ATP in the mitochondria. NADH dehydrogenase. So this is making you guess what that is. Dehydrogenase means to remove hydrogen. Yeah, so all of these are, this first pump is an NADH dehydrogenase because NADH is coming up here, dropping off its hydrogens. This is the first pump, and if you block it, same thing. You don't get any of this happening. Okay? Third one, oops, wrong way. Third one was FCCP. Now, if you put a hole, let's say you put a hole right here in this membrane, what will happen? what will escape? Well, fluid will escape out, right? But really, we're interested in this story because the hydrogens usually come through one route, this purple enzyme. But if you put holes in the membranes, the hydrogens are going to fall out or slip out from it, just passive diffusion, right? They will come out, and then you will no longer get the high concentration inside, so the hydrogens won't be coming through the ATP synthase, so you get no ATP. This, however, when you put a hole in it, this thing still happens, right? But the body is like, wow, we keep trying to make ATP, but it's not working. It's not working. Wow, we better use all of our glycogen and try to make, glucose into pyruvic acid and go through the steps. And then you run out of glycogen, so your body goes to the second place, which is adipose, where there's storage of lipids. And goes, okay, let's got to use the lipids. We've got to use lipids to make glucose, glycolysis, all these things. And you can see people would lose weight, right? Except it doesn't recover. You can't put a hole. Think about this. You can't put a hole in the mitochondria and then all of a sudden stop taking the drug and have the holes repair itself. Okay, that doesn't really happen. So unfortunately, it was a dangerous drug and it killed people, which is one of the worst kinds of drugs, right? You were trying to use drugs to help people. So, let's see, what else do I want to say about that? I think that's it. If you have any questions, let me know. So that whole part, the whole time I was talking, long time, was on carbohydrate catabolism. So now we're going to do anabolism. So we're going to build. Want to build the carbohydrates. So remember this picture? This is glycogen. Putting all the glucose molecules together is called glycogenesis. This is making glycogen from excess glucose. It's controlled by the hormone called insulin. Insulin is made from where? Where do you make insulin? From the pancreas, right? Pancreas makes the insulin during good times, all right? So we just ate. You're going to sit back, rest and digest, and you're going to take the sugar from what you ate, or the lipids turned into sugar, to put in storage. Stored, remember glycogen? Stored in the liver and the skeletal muscle. If you break down the glycogen, that's called glycogenolysis. So the lysis we've been through before. And that's controlled by the hormone glucagon. So don't get confused with the word glucagon and glycogen. I mean, glyco all means sugar. So there's that. It's on purpose that they picked that word. Okay, so this is perfect homeostasis story because you have two things working for negative feedback. You have insulin, glucagon. So when you're hungry, the glucagon is there. When the insulin, it's because it's good times and you just ate. Okay, and glucagon also made by the pancreas, right? This is to show you how you can watch this happen. This happens all day long in your body. Glycogenesis and glycogenolysis. Glucose comes in and it's converted into glycogen going up, or it's going to be used to make ATP. If you go down, it goes through glycolysis, citric acid cycle, electron transport system. When you're hungry, the glycogen needs to be broken down, so to the right here, it will come down and go to the left, and it will come out. So we got to keep blood glucose levels like around 100, 110. Those are the numbers. It can fluctuate, up and down. We have a range that's acceptable, and if it gets too high or too low, you get hyperglycemia, right? If you're too high and too low, it's called hypoglycemia. And most of you don't get hyper or hypo, you stay within the range, okay? Next, lipid anabolism. So let me look at this picture. This picture shows a triglyceride, so let's review this. Which part of this triglyceride is unsaturated? This has 3 fatty acids, right? One is unsaturated. Which one? Top one, the middle one, or the bottom one? The top one, yes. So if you don't remember saturated and unsaturated, go back, review that. To build fats from sugars is lipid anabolism. And the way it works is acetyl coenzyme A from glycolysis can be made into fat. Did you know that? You can actually take sugar and make it into cholesterol, triglycerides, and fatty acids. Amazing! This is called lipogenesis. The body is amazing. It has figured out how to make everything it needs, because a complete diet with everything you need in it has not been something that humans have had for very long. So there were good times and there were bad times. And during the bad times, your body had to make what it needed from the food that we were able to eat. So when I was in college back in the day, it was super popular to have non-fat. Non-fat stuff came and shook everybody up. We were all going to get skinny from eating non-fat donuts, non-fat baked goods, non-fat milk came around. Oh my God, it tastes awful, right? Non-fat milk, we're out, oh, it's like water, but we're going to get skinny. Did anybody get skinny on a non-fat diet? No, because to make it taste good, what did they have to put into the baked breakfast? Yummy. They had to add sugar. So if you look at the sugar, I may have mentioned this before, the sugar is too high in all of the non-fat foods. Way too high. It would be better for you to eat real fat food than to eat non-fat food with all the sugar. And this is why, because sugar is converted into fats. Okay, lipogenesis. Most of it is stored as white adipose, but I did want to introduce brown adipose to you too. So white adipose is where, it's not white, right? When you cut open a tissue, the body or an animal, it's actually yellow color, right? But we call it white adipose tissue, where the body stores most triglycerides used for making energy. This is a great way to store energy because it protects your body, right, from being bonked around. You have the fat there, and it can come and go. So you add it, you take it away. It's like the bank. Brown adipose tissue, I wanted to introduce to you because some people have heard of it. Some of you are taking exercise physiology. It is mostly found, though, in newborns and is used for making heat. And it stores a lot more calories. And if you think about this, it's important that newborns are born with this high amount of brown adipose tissue to make heat, because you never know, something happens to the mother, how's the baby going to survive, right? So this way they can live for quite a while on the heat that they make from their brown adipose tissue. But as we get older, we lose that brown adipose tissue and we don't have that type of storage. Alright, so we did lipid anabolism, now we do catabolism, and this is the breakdown of fats for energy. It's called lipolysis. This is real lipolysis. We're not doing liposuction, but lipo, right, means lipids, fats. The lipids are turned into, cut, right, into glycerol and the free fatty acids into its pieces. The glycerol, remember the CH3 molecule I showed you last time, that can be converted into pyruvic acid, which then can become glucose. So you can make sugar then from eating fat. And the fatty acid, which the long chains from the lipids, can be converted into acetyl coenzyme A, which then can be made into sugar also, into glucose. So again, just showing how your body can take whatever it has and turn it into the things it needs. If there is too much lipolysis, this results in something called ketone bodies. So the picture at the top right is showing you a ketone body. This is a breakdown of a fat into something called acetone. Yes, this is the same acetone that you would use to take off your fingernail polish. Okay, so as you know, that stuff is toxic. Well, ketone bodies in your body are also toxic. So if acetone is made, of course, that's dangerous. It can cause blood acidosis, which we'll talk about another day, but you can imagine that word. Acidic blood is not good. And this you won't see unless somebody is starving, which of course we don't see too often in our nation. But you can see this in diabetes, uncontrolled diabetes. So if someone has diabetes and is managed, you won't see this. But if they have uncontrolled diabetes, one of the symptoms they can have is blood acidosis from too much breakdown of fat, and they get the acetone. By the way, this is not so likely in a type 2 diabetic. This would be something you would see in a type 1 diabetic. So when we get to the endocrine system, we'll talk more about diabetes. The last one is protein. Protein, so we did carbohydrates, lipids, and proteins. Those are the three major nutrients. Proteins are usually not used for energy. Number one, your body's going to choose for energy is sugar because it can go straight into mitochondria. Fats are second because it takes a little while to break them down into the pieces that you need to go through glycolysis, citric acid cycle, electron transport chain. And proteins will be third. And this is because if you have to run from the bear, fight or flight, your body does not ever want to break down muscle tissue because it needs energy. Because you never know, it's the last thing that's going to go. And it doesn't usually happen unless someone's starving, right? And then the heart gets weakened and all kinds of stuff like that. So usually we eat protein to replace old amino acids. So good protein is recommended in your diet. Lots of it is good for you. Any extras, though, are going to be urinated out of the body as urea. So remember we did urea in one of the labs, right? So excess protein is not we don't store it like we do glycogen and adipose. So we eat it, and if you don't need it, your body pees it out. Okay? So it's released in the urine as urea. The amino acids, after you eat a protein, right, they're broken down into the smallest piece, which is an amino acid. That can be converted into pyruvic acid, and there you go. You can make it go into sugar, make it into fat. So who knew you could take amino acids and turn them into fat and sugar? But like I said, your body is a pro at being really creative and clever with our energy sources. So this picture is showing the interconversion of fat, protein, and triglycerides. So here you can take protein, you can get the amino acids, look, you can follow it this way, these arrows go back and forth, and you can make it into glucose and store it as glycogen. You can have fat, and you can store it as glycogen, you can store it as protein. You can put it through the citric acid cycle. All kinds of things. You get ketone bodies, lactic acid, it just goes back and forth. It's best for you to have a diet that has a well-rounded amount of protein, sugar, and fat. Okay, this of course assumes that the human body, right, is well adapted for times when you can't have all of those. The last slide is table 5.4, the relative importance of different molecules in the blood with respect to the energy requirements of different organs. Okay, that's a mouthful. Let's see. What is it really saying? It's saying these are the organs and this is what it prefers to use as energy. And you already know this. Brain likes glucose. So you need a little, wake up for your brain. Maybe you have a little sugar, right? Maybe a little candy snack, right? Something to give you a little bit of glucose. Fine. But take a look at your muscles. Did you know that skeletal muscles actually prefer the energy source as fatty acids? So does the liver. And maybe some ketone bodies. The heart likes fatty acids. So your brain needs the sugar. But the other things important to have in your body, some fat. So imagine going on like a no-fat diet. It wasn't good for our muscles, our liver, and our heart to do that. Okay? I think we're all done.